we are now broadcasting live. We'll be getting joined here shortly. Hello, everyone. Thank you so much for joining us today. Hello, Data Bees. Thank you for joining. And I'm curious to find out where everyone is joining us from. So if you want to go ahead and drop us your location in the chat, we'd love to see where you're joining us from today. We're just going to give it one more minute, folks, before we get started. We're letting the room fill up.
We have a lot of good friends here attending. Rita, who is part of the Hive.
Hey, Rita.
Who manages finance for the Hive.
Yes.
Jan Grottenberg, a good friend of ours, Curtis Abbott. Welcome, all of you.
Hello, everyone. Thank you so much for joining us today. If you want to share your location, please feel free. We love finding out where everyone's joining us from. Just go ahead and plop it in the chat. I'm joining from the East Bay area. Ah, nice. All right. Portola Valley. I could have guessed that. Okay, great. Awesome. So room is just finishing filling up here. We'll give it one more minute and then we'll go ahead and get started. Thank you everyone for joining us today. Joining from Los Altos. Awesome. Perfect. And can everyone hear us okay, audience members? Hi from Los Altos. Hello, Jan. Great. Awesome. Great. Very cool. Okay, we'll go ahead and get started here in just a moment. Thanks everyone for being here. We're super excited. We're going to have a great conversation. Menlo Park. Awesome. Awesome.
And everyone be safe. The fires, COVID, which we're going to hear more about.
Yeah. Coming in hot. Awesome. Thank you everyone for being here today. Welcome to the Hive Data Think Tank webinar series. I'm so excited to have everyone joining us from around the world. We've got an awesome panel today. It's really more of an in-depth conversation. Here we go. In-depth conversation. We're going to be talking about our CDC and FDA guidelines reducing the quality and quantity of COVID-19 tests with Nickhil, CEO of GenePath Diagnostics, and Sudnya, who is a fashion designer and founder, and she's going to be playing the moderator role. These two folks know all about COVID, all about testing, and I'm going to let them explain more in just a moment here. But before we dive in, my name is Maddie Watt. I am the Senior Manager of People and Programs at the Hive. I'm going to just lay out the ground rules for today's webinar, and then I'm just going to really quickly talk about what the Hive Think Tank does before I pass the virtual mic over to TM Ravi, the Hive's Managing Director and Founder. So, just ground rules. We love questions. Love, love, love questions. But to ask a question, please use the Q&A button. So if you look at the bottom of your screen, you're going to see a button right at the bottom, Q&A. Please use that to ask questions. It's just much easier for our panelists to see it right there in one area instead of looking there and in the chat. So if I see you ask it in the chat, I'm going to ask you to use the Q&A feature. So please just use that button. Also, you can upvote questions too. So if you see a question that you find really interesting, just go ahead and upvote it, and that will make it trend towards the top and has a better chance of getting answered on today's call. Please be respectful in the chat and refrain from product plugs. This is not the place or the time to recruit for money or for people for your startup. We appreciate you being understanding and working with us. This session is being recorded and it will be available to view later on the Hive's YouTube channel. I'll go ahead and drop that link for you guys in the chat. And we love when people follow and contribute on social media. Please use the hashtag HiveData or tag us at HiveData. And lastly, at the very end of the session, I will send out a very brief event feedback survey. It's like 5 questions, takes 2 seconds. If you have time, it's awesome if you fill it out. It helps us specifically design these events to fit your needs. So without further ado, what is the Hive Think Tank? We are an ecosystem of entrepreneurs, corporations, and thought leaders. Currently, we are a membership group of over 11,000 individuals. So thank you, Data Bees, for being a part of the Hive Think Tank. And I'm going to go ahead and drop our meetup link in the chat as well. So that's where you can find out what the latest events that we're doing. We're bringing you virtual events every week, and we're also bringing you guys content and thought leadership on what we believe in our investment thesis, and that is on our Medium page. So I will go ahead and drop our blog link as well. Next slide, please, Ravi. Thank you. Just want to do a quick thank you to our sponsors. They're coming up on this next page. Awesome. So we have all these sponsors here. Thanks to all of our sponsors. Love working with you guys. If you want to sponsor the Hive Think Tank, please get in contact with me. My email's down there, and I'll go ahead and post it in the chat. Also, if you're interested in doing an event with us, please go ahead and reach out to me. We'd love to work with you. Awesome, great. And then our last but not least, before I hand over the mic to Ravi, I just want to mention that next Wednesday at 9 a.m., so that's different than our usual 11 a.m. slot. At 9 a.m., we are going to be hosting an incredible panel featuring Odile Raoul, who is the founder of Fab Co-Creation Studio. She is amazing, and she happens to be an advisor and longtime friend of the Hive. And we also have these other fine individuals that are changing the world of fashion and luxury based on data-powered platforms. So we've got Kate, we've got Jet, we've got Jeanette, and they're all doing really awesome things in the world of fashion and luxury using data and AI. So without further ado, please welcome TM Ravi, and I'm going to just go ahead and turn off now. Thanks so much, guys.
Thank you, Maddie. And just a little more context on this event we're doing next week. So Odile has been at Chanel, at L'Oreal, was the president of Lancôme. And this whole topic of sustainability is something that corporations and different sectors are dealing with in the energy space, in the food supply space, and guess what, in the fashion area also. So I hope that you can join us next week. So just a brief introduction to the Hive. The Hive is a particular kind of venture capital entity, which is often called a venture studio. And our focus since 2012 has been on leveraging data and AI in the enterprise to drive transformation, to drive new opportunities, drive disruption. And you'll see sort of here in this slide, some of the themes that we are focused on, some of the technologies that our companies tend to leverage, and the industries that we play in. And so if you are interested in being an entrepreneur, would like to work in one of our companies, feel free to reach out to Matty or me. If you're going to be tweeting today, please use the hashtag HiveData so that others can follow you. And with that, I'd like to introduce Sudnya Shroff and Nickhil Jakatdar. So Sudnya is going to be moderating the event today. Sudnya is really a kind of a multifaceted individual. She was trained as an engineer, and in the early part of her career took a traditional path and worked at places like Intel. And more recently, she's been a writer, she's been a fashion designer. There's a connection to our event next week, and also an artist. Also joining us is Nickhil Jakatdar. Nickhil is the CEO of GenePath Diagnostics, with a presence both here in US and in India. And I'm sure he'll talk more about it. Nickhil is really one of the brightest, one of the top entrepreneurs here in Silicon Valley. Very early on in his career, as he was doing his PhD, started a company which had significant success, was sold to Tokyo Electron, has been a serial entrepreneur since then, and had great success. With that, let me hand it off to Sudnya.
Good morning, everyone. Thanks, Ravi and Maddie and the Hive for giving Nickhil and me this opportunity to bring the discussion to your think tank as well as open it to a larger audience. Um, COVID-19 has already been very deadly for the United States. The 1980 H1N1 Spanish flu, an unusually deadly influenza pandemic, claimed 675,000 US lives from February 1918 to April 1920 in 4 successive waves. 6 months into the lockdown as of this morning, COVID-19 death count on worldometers.info is at 182,826 in the United States, and this is still considered the first wave. What it translates to is 22% of the world's COVID-19 deaths, even though United States has only 4% of the world's population. Last Sunday, the White House and the FDA touted a coronavirus breakthrough that would reduce mortality rates by 35%, and it took just one day for the claim to completely fall apart and the FDA had to offer an apology. However, the damage got done. Similarly, earlier in March, the FDA rushed an EUA, or also known as the Emergency Use Authorization, for the malaria drug hydroxychloroquine, only to recall it not soon after. And just as of last night, NBC News reported that CDC has quietly tweaked its guidance on COVID-19 testing just this last Monday, making a change that could result in fewer people being tested and hinder contact tracing efforts. The change comes as political pressures continue to be at odds with experts who insist that we need more testing, not less. Today, scientific bodies like the World Health Organization, CDC, and FDA are being attacked by politicians in power, spewing ignorant rhetoric, which is only complicating our handling and recovery from this pandemic. It has never been more imperative than now for us to put on our critical thinking hats and arm ourselves with facts based in science, use therapeutics and treatments that have been studied and peer-reviewed, and most importantly, listen to medical experts and workers who are working around the clock on the ground to bring us life-saving solutions. With this in mind, I'd like to welcome Nickhil, CEO of GenePath Diagnostics, a molecular diagnostics research and clinical lab operating both in the United States and India. Nickhil.
Thank you, Sudnya.
Can you start by giving us the context of what brought you into this coronavirus battle?
Sure. I am not a medical professional by training. I'm an engineer by training. But about a few years back, when my mom was diagnosed with cancer, I got into the space just so that I could better understand how to get her the right treatment. And one thing led to the other, and I realized that the state of healthcare, whether it's the US or India, is pretty poor. And there's a lot of really great breakthroughs, but most of that does not reach the layman. And so how do you get knowledge, access to this knowledge out to more people? And how do you make these technologies, which are out there, great innovation that's out there, accessible to the common man? And so with that in mind, I joined a company as the CEO. It's called GenePath Diagnostics. It's been in business for about 10 years now. And their goal is to make molecular diagnostics, what sometimes is referred to as genetic testing, to be made accessible to everybody by making it affordable, by making it reasonable to get to not just the rich, but even to folks in the developing markets. And so with that in mind, we got started. And soon after joining, maybe 6 months into joining, COVID hit. And we were already creating tests for chikungunya, dengue, malaria. And so COVID happened to be one more infectious disease. And so we got into the business both of creating test kits for COVID-19, as well as running a clinical lab, where so far we have run about 17,000 clinical samples to date.
Okay. Lovely. It would help, I think, for our audience to be on the same page with the coronavirus vocabulary. So before we go ahead, I would love for you to give us a basic understanding of the SARS-CoV-2 virus and its corresponding disease, which is COVID-19. Just some fundamentals so we are all comprehending you at the same level.
Sure. I'll make sure it's not full of buzzwords. I'll keep it as straightforward and simple as possible. The SARS-CoV-2 is a RNA-based virus, not a DNA-based virus, and it's an interesting virus because the SARS and MERS that we've heard of in the past, they were also members of the coronavirus family. This one is interesting because it seems to have evolved in the way it can get to people. It can infect people very easily. Many of you have probably seen the visual of a SARS-CoV-2 virus. It looks like this ball with the crown above it, because of which it gets its name. These spikes that make up the crown are very, very efficient at latching on to the cells, the human cells. And once it latches on, it then uses some of the material from the cell to break itself open and infect the human being. And once it gets in through your respiratory system, it then goes through your body and does damage to different parts, all the way from your heart and your lungs to even what's sometimes referred to as COVID toes. And so it's a virus that is become very good at latching onto its host, and it may not be as deadly as the MERS. The MERS, once it latched on, was way deadlier, but it was just not very effective at latching onto people. The SARS-CoV-2, on the other hand, is very good at latching onto people. So far, it has been not as deadly, but because it's so good at latching onto people, even though the percentage of mortality may be low, the absolute numbers are high because of the number of people it has been able to successfully infect.
Yeah, that definitely does explain the word deadly, in the sense that we are seeing very, very large numbers compared to MERS, but it's because it is just so successful at infecting, so that there's just many more. So now that we know it's so good at infecting and it has actually figured out how to infect us on large numbers, testing seems to be a natural way of making sure that those who have it can stay isolated so that they don't further infect others. So knowledge is, again, knowing that I carry the virus is very important. So everyone by now, in 6 months into the pandemic, have heard about tests and testing. What are the different kinds of tests available on the market, and what are the ones that we should be asking for? Like, if you could lay out testing as in terms of what's available and also how they apply to us at different points of the infection.
Sure. There are 3 primary tests that are spoken about. There are more types, but the 3 primary ones that most of you have probably heard of. One is what's called the RT-PCR test. It's a molecular diagnostic test that is looking for the presence of the virus, either by somebody taking a nasopharyngeal, which is a swab that they put up your nose, and sometimes you feel like they are sort of scrubbing your brain. If that's the feeling you have, it's called a nasopharyngeal. They're going into the pharynx. Sometimes they just do a nasal swab. It does not go as deep in. It's less uncomfortable. Or they put something down your throat, which is an oropharyngeal. Again, they put it down pretty deep. When they do that, the objective is to get whatever virus copies that may exist if you are positive, and that is sent through the RT-PCR test, and effectively it's looking for those copies of the virus if they exist. Okay? So it's looking for active infection. The second test is called an antigen test. An antigen test is not looking as much for the virus as it's looking for, remember I spoke about those spiky things that stick out of the virus. Those spikes, they leave behind a protein once they get into your body. And the antigen test is looking for that protein. So it's another way of looking for active infection. Once your active infection is over, that protein is no longer there. And so both the RT-PCR test as well as the antigen test are looking for active infection. The third test is the antibody test. The antibody test is not looking for active infection. It is used typically to determine if a person had the infection in the past. Your body, your immune system tends to produce antibodies to fight infections. This has always been true. And it takes about 5 to 6 days on average for your body to build up the antibodies, for your immune system to build up the antibodies to fight the virus. And so if you did an antibody test, once you're infected and probably after your infection is gone, you should be able to detect the presence of antibodies. And so that's more to let you know whether you had it in the past. Okay, so those are the 3 types of tests out in the market right now. There are others called LAMP and so on, but they are not yet as mainstream. The other thing that's worth talking about is for the active infection, which is the RT-PCR test and the antigen test. The sample type is typically a swab, like I mentioned, but it could also be saliva. And you'll have heard more and more of a saliva-based test. The antibody test requires a blood sample because it's looking for antibodies in your blood. So those are the different sample types. The accuracy of the RT-PCR test is the highest. Okay? If a good test kit is used, you should get 90 to 95% sensitivity, which means we've heard this term false negatives. If a test had 100% sensitivity, it would have no false negatives. But there is no such test. An RT-PCR test is about 90 to 95% sensitive, which means there could be a few false negatives. The test actually takes only about 4 to 6 hours to run, and maybe less. But of course, by the time the results get to patients and consumers, it varies all over the place, and we can talk about that later. The antigen test is 70% sensitive, which means there's a 30% chance you could be a false negative, but you'll get the result in 20 minutes. Okay? And it's much cheaper. An antibody test, depending on the test, can be anywhere from 70 to 90% sensitive, which means there can still be false negatives. That's also a 30-minute test. So that hopefully gives the audience a general sense for the different types of tests, their accuracies, their costs, their turnaround times, and when you should use each of them.
There is somebody in the audience who just asked a question, and it might be worth leading into that right now, since it's related. The member asks, why are the tests only 95% accurate and not almost 100%? What prevents this test from being more accurate?
Yeah, I think that's a great question. So typically there are 2 types of reasons why you would lose accuracy. Theoretically, there's no reason, but practically there are a couple of reasons. One is how well the swab is taken. If a swab is not taken well, you have not really scraped. If you've noticed, and I've got a test done multiple times on myself, and I have found different people taking swabs follow very different protocols. Some, rotate it in your nose 3 times, 5 times one way, 5 times the other way. Some labs actually require you to give both a nasopharyngeal as well as an oropharyngeal swab, and then you combine the 2. So the better a job you do at swab collection, the better is the accuracy. That's one part. The second reason why you lose some amount of accuracy is the sensitivity of that test kit itself. There are probably at least 50 to 70 PCR test kits available in the market that a lab can buy and use. These test kits are sort of given, they're quantified by what is called a limit of detection. And what that really means is what is the smallest number of RNA copies that test can detect. Now, on paper, let me sort of try and illustrate this better. When you take a swab and you sort of swab your somebody's nose, you're going to collect billions of human DNA. and maybe 1 or 2 copies of the RNA, if that's all that there is. Okay, to be able to detect 1 or 2 copies of the virus amongst billion copies of the human DNA requires a test kit to be very, very sensitive. And a limit of detection basically tells a lab what is the minimum number of copies that a test kit can grab, can actually detect. Okay. And maybe because this audience is probably a little bit more technical, I'll jump into it a little bit deeper. If it's too much, please let me know. But the general simple idea of a RT-PCR test is because there is just 1 or 2 copies of the RNA, you can't detect 1 or 2 copies of the RNA within billion copies. So what do? What a PCR machine does is it goes through a sequence of heating and cooling. One sequence of heating and cooling is considered 1 cycle. And each time you go through this heating and cooling, you double the copies. You're sort of creating copies of that RNA. And so you do a second cycle of heating and cooling, and you double it again. And so after about 30, 20, 25, 30, 35, some number of cycles, if there was enough RNA to begin with, it'll have become big enough, large enough to be detected. And at that point, you call it a positive test. If there was no RNA ever, or if the RNA was very, very little, it won't amplify it to be meaningful enough to be detected. So that's effectively how it works. Now, if a test kit is not very sensitive, then it's going to take way more than 40 cycles to amplify it to a level that it can be detected. And the typical rule of thumb at most labs is if it's more than 40 cycles, it's a negative, right? And so that's one of the big reasons why there are false negatives, is because there's a whole host of tests out there, and they're not all the same. They differ in quality. And given some of the FDA evaluation or validation processes, which are not great, you get all types of tests out there, some of which are completely useless. And I would never want to use them in our clinical lab, and some which are really good. But that is one of the big reasons why you don't actually get 100% accuracy.
That leads me into a question for you. That is, how are these tests on the market? In an ideal world, all the labs that have developed tests would be checked by one central body, and the best test should be out for everyone, right? Like a best one with the best sensitivity, the best specificity. But right now, as I understand it, that's not the case in the world. There were problems getting into this pandemic, which resulted in the central bodies decentralizing test development, and that's when the FDA emergency use authorization started. So can you talk a little bit about the quality of tests, what that has done to the quality of tests on the market, why some people have been reporting that even RT-PCR tests, they've come back 5 times negative, and yet they had all the symptoms. Can you tell our audience a little more about what is the range of quality of tests, that just because it's an RT-PCR test, does it mean all RT-PCR tests are equal?
There are quite a few important points in that question. So let me try and address them. Number one, all RT-PCR tests are not equal. The FDA started off with the right intention, which is there is a need for good quality tests, and there's a need for more testing. And we need to have more vendors, more companies that can build these tests. And so typically the FDA required companies to go through a pretty comprehensive process. Now, if the FDA really followed that approach, it would lead to really good quality tests being approved, but it would take 6 months. And there was no time to do that. So what they came up with was this concept of this EUA, or emergency use authorization. And what that required is, it required companies to be able to do the experiments on their own, in their own lab, put the data together, send it to the FDA, and say, we have done these experiments in our own lab. Believe us when we tell you that they are really good, and here is the data for you to look at. And the FDA would say, okay, you know what? If you give me all that data in this format, in a week's time, I will let you start using that test in your lab, or you can start selling it to other labs. And so that was how it got started. Very soon, thousands of people started submitting applications. So the EUAs just dramatically went up. And what was initially meant to be a 1 week turnaround time promise, today, I know some of our tests are sitting with the FDA for 4 months, and there is not even an examiner assigned to it. And so they've been completely overwhelmed with the system that they were trying to put in place to speed things up. But that system has now got the worst of both worlds, which is it has not sped anything up, but poor quality tests are getting approved because the way that they're validating it is very questionable. And so the first part of the question, there are all types of tests. And the reason there are all types of tests in terms of quality is because of the process that I just described.
Is there any control I can have if I go to a lab locally or like, and say I want a particular test that I know has high quality, or is there no system like that in place?
No, you have no such visibility. The labs will, do high volume purchase deals with the various suppliers of these kits, and since they all say FDA EUA, the lab will say, you know what, if the FDA approved it, it's not my place necessarily to question it. But the really good labs do question it. I know, for example, in our lab, we have tested at least 15 different test kits which are used around the world. And even these highly used test kits, they vary all over the place. So while we know what our own test kit does, we in our own lab, because we also run a clinical lab, in addition to our own test kit, we have to use at least one other just so that we have a dual source. And we are just shocked by the quality of tests. I would never subject anybody I know to most of these tests because I know it's going to give a false negative.
So is that why Bill Gates recently called out the US on having failed testing? I think he had something to say about the timeline of results in addition to the quality, because he said, quote, "The majority of all US tests are completely garbage and wasted," close quote. What did he mean by that?
So I think one part of it is what we just spoke about, right? The quality of tests. But there's a bigger reason behind why he said what he said. The turnaround time on an RT-PCR test. I'll give an example just to to sort of provide the contrast and make the point. Because an RT-PCR test is meant to tell you whether you have an active infection, and you could always gain an active infection. If you don't have it today, you could get it tomorrow, right? So if you get yourself tested and swabbed today because you need to go, let's say you need to go in to meet the doctor, or you are having childbirth and so you have to go in to get admitted to the hospital, you need to get the result within 24 hours. In India, where we run one of our labs, if we do not provide the result within 24 hours, we will get fined by the government, and soon after we will get shut down. In the US, the average time to get a result is between 5 to 10 days, at which point it is completely useless for anybody to know 10 days later whether they had an infection or not, because the contact tracing is out of the window. If you were to get admitted into the hospital, you can't do that anymore. And so in general, one of the, I guess the number one reason why he called it a completely garbage is because there is no value in getting a result 10 days from now.
Makes sense. Another follow-up question, and there are a lot of questions that I think I will also leave towards the end if they are not, but if they are relevant to what we are talking about, I'm going to pull them in. Another attendee has asked, what stopped the companies from doing due diligence and only send to FDA the tests that they are convinced would work for their own family, let alone other people? FDA can do additional checking, but why the burden of proof is not on the companies themselves?
Yeah, I mean, that's how it should be. I know there are companies that do fall in that category, but there are enough for whom this is maybe an opportunity to make money. And, think about this. If Theranos were alive today, if it started today, they would make a killing because it sounds great. And there are companies like Theranos who will abuse the system if given a chance. So the question is right on in terms of how companies should behave, but is that the way they do behave is questionable. And the second thing is, many of you may have been aware of that famous Abbott case, that rapid test, 5 minutes. It was in the news. People said this is going to change the world. And yet 4 weeks later, after all that big deal was made, it turned out that 48% of the results from the Abbott test did not match what those researchers at NYU saw with other FDA-approved tests. That, I mean, if you have 48% of the results being wrong, you might as well flip a coin and decide whether you have COVID or not.
Right.
And this is not a fly-by-night company. And so I'm not sure what exactly is the reason why people don't do their due diligence in a better way. But there are enough data points to show that it's not happening, and that is actually really dangerous.
And then there's one question related to the swab, accuracy of the collection, like the way it's collected. Would taking 3 RT-PCR swabs actually improve the accuracy? I know we were short on swabs around the world, so 3 swabs would be a lot to reduce the 5%. But what are your thoughts?
So I think at that point in time, so for example, in our lab, we take 2 swabs. And now we're adding a third input, which is saliva. So we take 2 swabs and saliva.
2 swabs for what?
So nasopharyngeal, oropharyngeal, and a saliva sample. Okay. And that way we have effectively 3 inputs, right? To improve the reduce the chance of error, right? At that point, we'd be limited only by the sensitivity of the test kit itself, the one I described earlier, because the sensitivity of the test kit depends on the primers and the probes and the master mix, the materials you use. But if you had the best test out there in the market and you took more inputs like nasopharyngeal, oropharyngeal, and saliva, you are basically going to get closer and closer to that 99.95%.
Looking forward, can you tell us where innovation?
Sorry, can I make one more point, which I think is important?
Sure.
When you take the swab is also important. So what data has shown is from the day you get the infection, the day the virus enters your system, it takes about 3 or 4 days until when the tests are not as sensitive, because the virus has isn't enough of it. When there's enough of the virus and your symptoms are increasing, or if you're asymptomatic, it's not increasing, but you just have enough virus, at that point in time, you will start with a meaningful enough amount of virus particles or copies that come on your swab. And that is true for about 5, 6, 7 days after that. After some point, the amount of viral material starts dropping because you're losing your active infection. At that time, again, if you try to take a swab, it's not going to give you a large number, which is fine, because at that point, it's okay. But those first 3 or 4 days, from the time you started getting the infection, getting the virus into your system, to the time you start developing symptoms, your accuracy of your tests will not be as high.
Okay. I wanted you to expand a little bit on the innovation in testing that will make these challenges that we are discussing easier to overcome, because it looks like we have a long way to go in this pandemic, and we are going to really need a robust testing system so that we can then have a robust contact tracing system and eventually get this pandemic under control before we even think about elimination and eradication.
Sure. I think there are different aspects to innovation. Like, where do you want to innovate? You could innovate on reducing the price of the test so that it becomes easier to run it multiple times and you can make it affordable to more folks. So one is around the price. Second, innovation could be around the turnaround time. Today it takes, like I said, the actual test only takes 4 to 6 hours or less, but results take 5 to 10 days. So there is, a number of things that are probably happening behind the scenes on the logistics and supply chain side, rather than on the actual test aspect. So there is innovation around the turnaround time. And then there's innovation around getting it closer to point of care. So for example, let me give you examples of each. On affordability, how do you make it cheaper? There is one of the innovations that's been actually touted by the FDA as something that they are looking for, is something that impacts both price as well as turnaround time. And that is the concept of pooling. Okay? where effectively you pool 4 samples or 5 samples together, okay, and test them. If all 5 samples were negative, then you don't have to sit and test each of them individually. If even one of them is positive, then you will have to look at breaking up and testing each sample. This approach works really well when the incidence rate is sub 5%. Okay? But if the incidence rate is very high, then pooling is not going to help much. There are other smart algorithmic approaches where you don't even have to break it up. You arrange your test in such a way that you can pinpoint without doing the next level of testing. But pooling helps reduce turnaround time, right? Because now you're doing 3-4 times higher number of samples at the same time, and it reduces the cost. Right? So that's an example of one thing which helps over there. Another example which has now started hitting the market is saliva. Saliva will allow or enable people to provide their sample where they can collect their own sample, because doing your own nasopharyngeal, oropharyngeal is very hard. But spitting into a tube and shipping it out is much easier. So it makes it more accessible to a lot more people. And it can also reduce the cost, because then you don't need to have people sit and collect. So all the people costs of collection go down dramatically. Then there are innovations in the actual process. So there is the PCR step. And before the PCR step, there is what is called a sample prep step, where you extract the RNA from the swab or from saliva and put it into the PCR step. This ends up being one of the more time-consuming steps. And now there are innovations out there that have to do with getting rid of this step almost completely. And I can talk about these because we are involved in all of this. We have got EUAs out to the FDA on each of these topics. And there are others as well who are doing amazing work. But of course, if it does not see the light of day, these are innovations that are going to sit around without actually being practical. But there is a lot of really amazing work happening in a number of different areas, which can reduce price, can reduce turnaround time, can make it more accessible. One of the more interesting things that is happening, and I referred to it earlier, was this thing called a lamp-based test, where in a PCR test, I spoke about how you cycle through the temperature, up and down, up and down, up and down. That makes the equipment a lot more complex. With a lamp-based test, it's what's called an isothermal reaction. You can do this on a hot plate. You can do this at point of care. You can have this outside a company. You can have it in a village. And this test takes about 10 to 15 minutes to run. It can be run on cheap equipment. It can be run at point of care. And you can run it every day, which becomes a very good technique to sort of get more people tested on an ongoing basis. It's not as sensitive. But when you are in the mode of trying to monitor, then this type of test is perfectly okay. In fact, lamp-based tests as well as PCR tests with pooling are two of the techniques that are being used by many of the universities. The bigger universities that are opening up right now, they are actually Penn State, University of Michigan are examples of schools that are going down the path of one or both of these techniques, so that these 50,000, 100,000 students, staff, faculty can get tested 10,000 every day. And that's a scale that requires these type of techniques.
Thanks. You've quantified time for us, like speed for us, and compared. Can you give us an idea of what do we mean by cost? What do we mean by cheap? And what do we mean by expensive? We haven't really touched on that briefly. If you could just put that in.
Sure. So an RT-PCR test today in the US is reimbursed by insurance for about $70 to $100, depending on a few factors. The same test in a market like India, where it's not really insurance driven, it's more out of pocket, started off, the government mandated it to be no more than $65 when it started. Within 3 months, that $65 has been now brought down to $28. And within the next 2 months, it will probably be a $15 test. So all these innovations are causing the cost of the test and the price of the test to go down significantly. To me, one of the things that's unfortunate is in a market like India, you can get at this point a $20 test for RT-PCR in less than a day. in the US, that same test costs $100 and takes 5 to 10 days. And there is something fundamentally wrong with this. And Bill Gates, I know, had a recommendation, which is insurance should be or lab should be paid based on the turnaround time.
And then that would take care of them making sure that the higher those who pay for it actually get the answer in time. That's what he meant.
No, even if it's insurance covered, if the lab gives the result in a less than a day, the lab is reimbursed at the $100 price point. If they did it in 5 days, pay them $0 because it's useless.
That's right. Have you observed or recorded recurrence of COVID-19 in your tested patients now that you have had 6 months of experience and 17,000 plus samples? Because that's another concern. And then there is a question in the Q&A that also asks, how does a person know how long since they have been infected? Does one need to get tests multiple times to make sure that they hit the right time period suggested by speaker?
Yeah. So the first question was around can you repeat the first question again?
The first one was about, have you seen recurrence?
Yes.
And
yeah. So we have done now 17,000 patients to date. And because we work very closely with the doctors, we find out how many of them, recovered, how many of them did not and passed away. And so we have all these different cases. But some of the most interesting observations we have had, and in fact, it happened twice with our own employees, is that there was an example where one of our employees tested positive.
And your employee, you mean what kind of employee? Because you have a test lab, and so you have people in the clinic and...
Yeah, it's an employee for our test lab. And he was... whenever somebody starts with us, we first get them tested. So he was starting with us, we got him tested, and it turned out he was positive. And so he had to be quarantined for 2 weeks. He was young, didn't show any symptoms. So the recommendation is to be quarantined at home. He came back 2 weeks later. We tested him again. He was negative. We tested him with the antibody test after that. Right after that, he was positive. So it all made sense, right? Then 4 weeks later, he complained of symptoms, and we tested him. He was again positive. We tested him with RT-PCR test. He was again positive. We tested him with 3 different RT-PCR tests, just to be sure. And he was positive by all 3. And this time when we tested him for antibodies, he was negative. So in his case, in 4 weeks, his antibodies disappeared. Okay. Now, is that a one-off? Does that happen 5% of the time, 80% of the time? We have no idea. But now we have seen about 6 to 8 examples like this, where people have had recurrence. Now, we have also seen other examples where we used a different test. They tested positive the second time as well. But the second time, they tested positive for H1N1, okay, not for COVID. But the symptoms looked very similar. And so very often when at this point, because the focus on COVID-19 is so high, if doctors see certain symptoms, many of them assume you have COVID and they ask you to quarantine. But maybe you don't have COVID, maybe you have H1N1. And so it's critical that you actually get tested with the right tests to make sure it's one and not the other, or vice versa, right? So
it's going to be important for our doctors to not only test us for coronavirus, but a panel of viruses, because our symptoms could present themselves. So that's something we could go and ask our doctors, should we be in a situation like that, right? That's really good information to know, that even if the COVID test comes back negative and I have symptoms, I could have influenza, and a Tamiflu would help, like a therapeutic that's based on the actuals, which could be missed right now because everyone's so single-minded.
So what's happening right now, because of a lack of testing, very often doctors right now, if there is a problem, they will prescribe Tamiflu, they will prescribe antiparasitic, antiviral, they will prescribe dexamethasone for a cytokine storm, just in case your immune system kicks in and starts going crazy, which happens to a few people, and they will prescribe remdesivir. And in India, they also still prescribe small amounts of hydroxychloroquine. So right now they're sort of saying, look, it's going to take me a long time to get the test results, and it could end up being something else. So let me throw the kitchen sink in terms of the treatment side. If I can get tested, great. But if I can't, they're still throwing the kitchen sink at the problem.
What you mentioned about the antibody tests, how it is post-infection, but that data is really important in development of vaccines, right? The information we get out of how our bodies react and fight this particular virus and create antibodies, and how long those antibodies last, is information that goes into developing vaccines. Now, it's worrisome that you are seeing cases of people who get it so soon after. So, I know this is still very early in the research part of just this entire disease, but what do you think is the implication of this information going forward into the vaccine development? There's so much pressure in a vaccine being produced ASAP. Is that even a good idea to rush something like that so quickly?
So I think right now there are quite a few vaccines in the phase 3 of the trials. And phase 3 is where they actually test this on the 20, 30, 40,000 people. This is when the real data will come out as to whether it's going to be 70% in terms of its efficacy, 50%, 95%. I know Dr. Fauci about a couple of weeks back said that if the vaccine comes out with 70% efficacy, he would be thrilled, right? And so what that means is that for 30% of the people, it's not going to be effective. That's one issue. And the second issue is, when surveys have been run, a lot of people are nervous, and these are not anti-vaxxers. These are regular people like you and me who are nervous about taking the vaccine so early when it's been rushed through. And of course, the third issue is that the number of people who will actually get access to the vaccine in the beginning is going to be limited, right? First, it's going to go to the higher risk patients and to potentially military, healthcare. By the time it gets to people like us, it's going to take longer. And by the time it gets to people in the poorer countries, it's going to be a long, long time. And so the combination of all these three things, whether it really, what is the efficacy, how many people are actually going to take it, and when is it actually going to be accessible to the layman? These three together tell me that we are still talking about, at least a year before we have any sign of seeing whether it's working or not.
Then with that in mind, knowing that at least a year out, testing and being aware of our health and the health of people around us is going to be the most important thing. What are the basic steps that you would suggest we all should be aware of and continue to follow now that we're going further into the flu season?
Yeah. I mean, some of the things are actually pretty straightforward, right? I'm just always amazed why wearing a mask is met with so much resistance. It is one of the most effective ways. Washing your hands with soap and water is, again, very simple to do. And so I think those are basic hygiene. I think the issue I see is, because this has been going on so long, people are getting tired of the virus. Unfortunately, the virus is not getting tired of us. And as people get tired of the virus, they're almost sort of wanting to latch on to any sign of good news. Anything that sounds like it's promising, here comes our savior and the world is going to go back to the way it was earlier. And I just don't believe any of that. And so I think just follow basic practices. Don't, take social media too seriously. And personally, if I'm ever in doubt about things, I go to a site like the Mayo Clinic. Earlier, I would go to the FDA or the CDC sites. Today, I consider them pretty worthless because they themselves change their views within 24 hours. Sites like the Mayo Clinic and others like them, Johns Hopkins, their sites typically end up being a lot more believable, credible, and trust that over lots of other things.
In the interest of time, I'm going to jump into some of the questions. There's Apurva Desai asking, are public health and environmental conditions such that new coronavirus strands are more likely to occur more frequently in the future? He's basically asking, should we be prepared for a COVID-22, 25, and all the work that you're doing for COVID-19, would that form a good foundation for, will it prepare us for some more coronaviruses?
Yeah, I think this has got the world on its guard. I mean, nobody expected 6 months back that we would be here. This is the type of stuff you read in books and watch in movies. Now it's happened, and I think it's led to us hopefully being a lot more prepared for the future. A lot of the work that has gone in this to do this testing, create these test kits, will be applicable, will be useful even when if it comes up again. And I do believe this is not the last we have seen of a deadly virus, simply because viruses are evolutionary beings. They are going to keep evolving. I mean, what did SARS-CoV-2 learn from its predecessors? How to get more effective at latching on, right? Each virus, I mean, human beings, because our lifespan is, 50, 80, 90, 100 years, we have to go through thousands and thousands of years to evolve. For a virus, the lifespan is so much shorter that they go through their evolution within months. And so in a few years, is this virus and other viruses like this going to learn and evolve? Absolutely. And so we better get better and better at handling it. If this experience hasn't taught us, I don't think anything will. So yes, it's going to happen more often. We have the foundation to build on, and I hope we do.
Curtis Abbott asks, I hope you will talk about CRISPR-based testing somewhere along the way. I don't know if we have time, but I just want to address that question.
Yeah, I mean, I think that's a whole topic that's worth a full conversation. I think at a high level, I would say that CRISPR-based therapeutics, I actually believe is more real. CRISPR-based diagnostics, in my very blunt opinion, is more buzzword compliant than real. So CRISPR is powerful. I think its use in therapeutics is real, and I think the world is going to transform because of it. CRISPR-based diagnostics, if you look at the details, it's basically a PCR run followed, like a LAMP-based run, followed by some CRISPR, which if you didn't have it, nothing would change. So maybe this is controversial, but I don't think CRISPR-based testing is promising, but CRISPR-based therapeutics very.
The next question is, which tests are the best, most accurate with the shortest turnarounds, and what companies develop them? This is like that question which all of us have in our mind, and we wish we could go to the counter and say, I want that test. But what can you tell us more?
I mean, there are a few tests, and I can tell you this based on our experience. This is by no means meant to be comprehensive, but obviously the GenePath test has been tested. It's shown to be one of the most accurate in the world. And that's why CLIA labs in the US are switching over from what they were using earlier and over to our test. So that's a thing, and so it's a shameless plug. There's a company called Thermo Fisher. Their test kits are also considered very good. There is a Korean kit called Lab Genomics, which is pretty reasonable. I would say, and there are maybe another 3 or 4 that are maybe one level lower that I would use in my lab, or that we do use in our own lab, because if we can't use it for our family, we would not use it on anybody else. And they cost, and these test kits range anywhere from $10 per test to $20. But then when you add in all the other overheads, that's why the cost of the actual test ends up being between $20 in India and $100 in the US. But the actual kit that goes into the testing is actually at this point between $5 and $20. Ours is closer to $5. Most of the other tests are between $15 to $20.
Yaman Kaushik asks, especially in India, health medical insurance companies only insist on checkups and tests at the time of policy purchase, but do not facilitate any such routine at regular intervals. Do you think it is time that insurance companies drive the diagnosis as part of their product and make it mandatory so that early diagnosis can help life recoveries?
I mean, this to me should be a very simple business case for these insurance companies to do. And that should answer this question. I think one of the reasons why it's not being done is because the price of these tests were kept pretty high. But if we can truly achieve this $10 and $15 test, not test kit, if the total test, instead of costing $100, can cost $10, and I believe it can happen in the next 2 months. If you did enough volume, there is no reason why a test should cost more than $10. If that happens, I absolutely believe that insurance companies, corporations, universities, everybody will say, we need you to do this test often.
Prevention. Mm-hmm. Are population monitoring tests that measure city sewage also using PCR to look for RNA?
Yes, there are quite a few programs, cities that are doing this. It gives you a general sort of relative view rather than an absolute view, but it is being done by a few cities.
Rekha Pai asks, because vaccines general availability will take so much time, do you envision these saliva-based, low-cost, low-sensitivity, high false negative tests becoming the default way we are going through with the next few years?
Actually, saliva is turning out to be pretty sensitive. So it can get to the 90% sensitivity levels. And the NBA, as some of you may have heard, the NBA is using these saliva-based tests. And so I absolutely believe that is the future.
Thanks, Nickhil. Thank you for bringing us up to date with everything going on in your world, and profound thanks to your entire team for their perseverance and a marathon effort as we race against time to save human lives. With that, we are just a little over our time, so I'm going to hand it over to Matty and Ravi. Thank you, everyone.
Thank you, Sudnya, for excellent moderation, and of course, Nickhil, for this great conversation. I want to thank my colleague here, Matty, also for putting the Hive Think Tank together.
Thank you all for joining us. Thank you to our amazing speakers. Thank you, Ravi, for always helping put on great events. Love everything that we're bringing for our community. If you guys want to fill out the event survey, then you can go ahead and give us feedback on what you want to see more of. I already dropped that in the chat. And then also join us next Wednesday, 9 a.m. Pacific Standard Time. Thank you so much. Stay healthy, stay well, stay safe, stay not on fire. All the stuff. Stay masked. Awesome. Thank you, everyone.
Thanks,
Bye.
Thank you, everybody.